Good evening, everyone. In the previous video, we talked about questions asked by the developmental biologist. And in this video, we try to answer those questions partly by using model systems. So let us begin. Organisms as model systems. To answer the questions that developmental biologists ask, researchers need a tractable experimental organism best suited to their questions. What makes an organism a good model for addressing a given question? Some of the common considerations in choosing a good model system are the following. Size. A particularly practical consideration is the size of the adult organism. Is it easy to house a significant number of the breeding adults in the allotted laboratory infrastructure? For example, housing 50 mice in a cage requires a lot more space and expense than housing 50 flies in a vial. Second important consideration is generation time. How long does it take the organism to complete its life cycle from embryo to reproductive adult? Additionally, how short is the embryonic period? The roundworm C. elegans has a full life cycle of 3 days, whereas it takes the zebrafish about 3 months to go from egg to egg. However, early embryogenesis in zebrafish spans only 24 hours. Next consideration is embryonic accessibility. To study embryology, a researcher needs to be able to see and work with the actual embryo. Different species pose different challenges for embryonic accessibility. Some embryos are dispersed in the water for easy collection, while others develop in an opaque shell, such as the avian egg, or in vitro within the womb, or as with mammals. Next consideration is feasibility of the genomic interrogation. Since Mendel's work with peas, developmental biologists have been driven to identify the genetic basis underlying all developmental processes, from embryology to disease. Although all life is based on the organization and use of the 4 nucleotide bases, no species has the same genome. Genome size, organization, and content all differ, which can affect the level of genetic interrogation that is possible. For instance, researchers studying regeneration in the Mexican salamander have to deal with the largest genome ever sequenced. Maybe the secret of the regeneration lies somewhere in all that DNA. Organism type and phylogenetic position. Ideally, the research question should guide the selection of a model system. If researchers are interested in the remarkable process of metamorphosis, then clearly they are limited to a select few model species that display such transformations, such as the fruit fly or frog. If they are passionate about studying human development, they may use a mammalian model organism, such as a mouse or human cell in culture. If their questions are focused on deciphering the developmental changes fueling evolution, they can choose species that occupy informative phylogenetic positions, such as the charophytic algae that are basal to multicellular land plants. Next important consideration is ease of experimental manipulation. Last but certainly not least, among the considerations is whether an organism is appropriate for the experimental approach needed to answer the question being asked. For example, due to the long history of the significant investigations to develop the fruit fly and mouse model systems, a plethora of powerful molecular and genetic tools exist to manipulate gene and protein function during embryonic development of these organisms. Similarly, the extensive body of the information now available on the genetics and development of the small mustard plant Arabidopsis thaliana has made it a widely used model organism in research on the flowering plants. Some of the more common model systems used to study embryonic development include a flowering weed, we have Arabidopsis thaliana, we have sea urchin, we have sea skirt, we have fruit fly, we have then roundworm and C. elegans, we have zebrafish, we have African cloud frog, जिसको हम Xenopus laevis कहते हैं. We then have chicken, we then have and lastly we have mouse, musculus. Advances in the shared genomic and molecular approach have dramatically increased the accessibility of non-traditional or non-model organisms for developmental research. This is one of the most exciting things about being a new student entering the field of the developmental biology today. You do not have to be restricted to the conventional model system. Rather, any species could be a new model organism for you to investigate. Thank you.